There's going to be a lot of people dying from bacteria if we don't figure out how to deal with them, because they're getting past our antibiotics all the time now. There are bacteria now that are becoming resistant to our last resort antibiotics. So if you don't respond to antibiotics, they put you in a hospital and they have some last resort antibiotics like vancomycin or linezolid, and many people die of this. Sepsis, MRSA, a lot of people die of this. It just kills so many people, and the worst place to get this is in the hospital. You say, in the hospital, that's where you get good care. No, what happens is the bacteria in hospitals are much more resistant. You're much better off being at home, and you get less exposed to these highly resistant bacteria. So last time we talked about single molecule nanomachines, these molecular machines. And now we're going to really get into the drilling through biological tissue. It's a new modality of therapy, and we're going to look at it from the sense of medicine. So this is really the first lecture we're having on using nanotechnology for medical applications. So we had looked at this one slide a little bit before, but you see that this will drill. When this starts spinning at these 3 million rotations per second, it drills through, puts holes in it, and you see this blebbing that I think we may have mentioned previously, this opening of the cells, and it ends up killing them. And so the way we studied this, you know, what you do, and this was work that we did with collaborators. We collaborated a lot. This is with Gufen Wang. One of the things that we do, and I sent a student to NC State at the time, is that we work with a lot of people that have many types of expertise. So we synthesized this molecular machine that has appended to it strong fluorophores, things that you excite at a certain wavelength and they will emit light at a different wavelength, usually at a lower energy. They'll emit light. And so the idea was that we would put these in just a vesicle. So this is not a cell, this is just a vesicle, a lipid bilayer, which you can make synthetically by just taking lipids and you put them under shear, and where it rubs them back and forth, and it'll form these vesicles. But inside the vesicles, we put Bodipi, which is a dye. This is a dye. And then, so we're watching the nanomachines with one wavelength. We're watching the Bodipi with another wavelength by watching where these two different things fluoresce. So we have the molecular nanomachines, and then we have the Bodipi dyes in there. And if these are inside and drilling on out, as they drill on out, they'll form holes. And as they form holes, the Bodipi should leak out. And so that was the test. And so that worked. You could visualize these, and you could see that the Bodipi was lighting up and the nanomachines were lighting up. And so what they're going to do here is they're going to track these. And so say we look at this one here. So just focus in on this one. And you can see it in these subsequent slides. And you're looking at the intensity. And once you start shining the light for 5 minutes, 10 minutes, 15 minutes, you start shining that light. And the intensity of the Bodipi signal will get lighter and lighter and lighter. So you see the intensity, if the original is 1, the intensity is dropping over the time of exposure. And some people will argue, oh, well, that's just because the thing heats up and it's leaking out. Well, that's not the case, because we put in one of our nano cars, interestingly enough, which actually has a larger absorbance and a larger extinction coefficient. And when you do that, you get very little leakage. So it's not the light that's doing it. And so that just begin to give a little bit of information. This work we did with Jacob Robinson's group here at Rice. And the idea was to do something called patch clamp. And that's an old technique, but what you do is you put a pair of electrodes, you put an electrode inside to a cell, and then you put an electrode over here far away, and you're measuring the current between that cell and the outside electrode. But if the cell is nicely closed, no current is getting out, because no ions are able to get out of it. And so we used a bunch of control molecules. This control molecule doesn't even have a rotor on it. So that's number 5. So if you take number 5 with no light, nothing happens. If you take the the molecular machine with no light. Nothing happens. There's no current transferring. But now if you just apply UV light with no molecules, again, no current is transferring. But if you apply UV light with 5, again, no current transfers. But if you apply UV light with the molecular machine, this is going to start rotating. And after a few minutes, so this bar here is 30 seconds, you started applying UV light here. So after a few seconds, now you start seeing current start transferring between the electrode that's inside the cell and the electrode that's outside the cell. And that means the cell membrane has opened up. If you do not believe in the physical resurrection of Jesus Christ, send me an email, tour@drjamestour.org, and we will get together and I will share with you about why I embrace the resurrection of Jesus. The other thing that you can see, there's blebbing on these. So you see this little blebbing here. There's a little bubble. Here's a little bubble coming off of this, and that's the innards of the cell busting out. Again, you have this propydium iodide in the cell, and it around the cell, and when it gets in, interacts with DNA and RNA, it'll start getting on in there. But and then we show this effect. I'm not sure I showed you this. This is where we can show that we're actually drilling right into the skin of a mouse. So these are like little molecular drills, and if you put the fast nanomachines on there and you shine light, you see it's just drilling right into the epidermis of the cell. Where if you take the slow nanomachine and light, you don't see any drilling in. If you just take the solvent that we use and light, you don't see any drilling in. So this can really drill down into the cell, into the skin. And then we change the molecular structure such that these would absorb visible light. And so when we put this nitrogen on there, it shifts the spectrum, and instead of peak absorbance around 360, it moves it to 395. And that means at 400, as soon as you're over 400, that's the visible region. That's where your eyes begin to pick up a signal. And so we're using the visible region, so the visible region starts right at about 400, and you still have a lot of absorbance at 400. I think we were using an excitation wavelength of like 405 nanometers, and so you still have a lot of absorbance there. So these can be used in the visible region. And now we applied this to cancer, and so cancer is the second leading cause of death worldwide. 10 million people a year die from cancer. And it's projected to be 16 million by 2040. The total annual economic cost of cancer is 1.16 trillion. 40% of men and women will be diagnosed with cancer at some point during their lifetime. In 2018, there were 18 million new cases and expected to rise to 30 million by 2040. And it's generally, cancer rates are highest in countries whose populations have the highest life expectancy. So it's predominantly, not exclusively, but predominantly age-associated, that have the highest life expectancy, education level, and standard of living. And you say, well, what does that have to do with it? Well, a lot of times those are more advanced cultures, and so they detect cancer. You know, you go in and you get it analyzed. If you're in a, living out on an island all alone, I mean, there's nobody to detect the cancer for you. And so, you just died. I mean, the person died. I mean, they don't really have a cause of death. And so you can see what happens when this is in pancreatic cancer cells, that if you take the fast moving one, this is a fast moving one, and put it on here, you shine light, and when that light goes on, it starts drilling into the cell. Propydium iodide gets into the cells and starts staining them. And you can see the fastest molecules, the fastest molecules, this will start happening. So in this case, it's just showing 5, and this is with 7. This is with compound 7 and light. You see that you get the highest in there. When you go to a slower machine, a little bit slower, or a little bit less effective, it's here. And then as you slow the machines down. So you do it with controls that don't rotate very fast at all. So this is less than a revolution per second. And so this has very little effect. And so you can see this killing melanoma cells in a dish. So the first thing you do is you practice this on just cells in a dish before you go into a rodent model. And so if you take 16,000 cells and you put it in this little container, and then you add the molecules, you still have your 16,000 cells. If you just shine light, you still have a lot of cells. There may be a little bit cell death, but not much. If you take this molecule, M96 plus light, boom, all the cells are gone. So you see that there's this cell death. So it's now if you have 1,600 cells per well, again, you're killing this. And so only when you have the molecules plus light do you get the very efficient kill. The number of surviving cells goes way down. Then you have, here's light-activated nanomachines, these same nanomachines that were applied to a tumor model in mice. So what you do is you take mice, and you can have two types of tumors that you can inject them with, two types of cancer. You can inject them with a mouse cancer, or you can inject them with a human cell line of cancer. And generally what you'll do is you'll shave one of the hips, and it will get injected here at the hip. This mouse is anesthetized. This is a gas chamber right here that you put over their nose, and it will knock them out. So they're not feeling any pain here. And that's a very important thing to do. There's really high ethical standards with these. You have to go through a lot of approvals. In fact, some people argue that there's more approvals for animals than for people. And the reason for that is people can sign away their rights. Animals can't. So animals have an advocate. And you have to make sure that you get so there'd be an animal advocate on the committee. And so you have to make sure that you have certain controls that are acceptable standards. And one of these is that you don't want to cause even stress to the animals. So you just take them and you put this cone over their nose and mouth, and then they immediately go to sleep, and you just keep it there while you're doing the experiment. And so what you do is you will allow this tumor to grow, and then at some point you'll inject, the students inject some of the molecular nanomachines into this tumor and then shine light. So they'll shine light on it, and they'll see how much does the tumor growth get inhibited through getting the molecule plus light. And you can see the tumor unchecked is going to grow at this rate. So be 500 cubic millimeters over a period of 15 days. That's how fast that thing is growing. When you have the nanomachines in there, it's growing less, because what it's doing, it's drilling into the cells. Now the problem with this is it's visible light. Visible light is safer than UV light because it's not as degrading to structures in the cell. It's not as degrading. But what happens is that when you use visible light, there's only about half a millimeter penetration into the skin. So visible light doesn't penetrate very much. UV light penetrates even less into the skin, because UV light is a more damaging light, so our skin is protecting us. So what we would have to do to have really good penetration is go to the near IR. The near IR would have to be, and then I'll show you the ones that we have made for the near IR. And so before we get into that new structure, let me show you the same molecules that we were using, but now not killing cancer cells, but killing bacteria. And so these are called these superbugs. These are bacteria that are not responding well to typical antibiotics. And these superbugs are slated to kill 10 million people a year by the year 2050. So like when you guys are around my age, it will make COVID look like a every year sort of event. There's going to be a lot of people dying from bacteria if we don't figure out how to deal with them, because they're getting past our antibiotics all the time now. And so there are bacteria now that are becoming resistant to our last resort antibiotics. So if you don't respond to antibiotics, they put you in a hospital, and they have some last resort antibiotics like vancomycin or linezolid, and these are very expensive. Like linezolid is extremely expensive, tens of thousands of dollars per treatment, and you try to knock these things out, and many people die of this. Sepsis, MRSA, you've heard of MRSA. A lot of people die of this. It just kills so many people, and the worst place to get this is in the hospital. You say, in the hospital, that's where you get good care. No, what happens is the bacteria in hospitals are much more resistant, and you're much better off being at home, and you get less exposed to these highly resistant bacteria. And here's the life of an antibiotic resistant. Here's what happens. So here's a normal bacterium. Here's a resistant bacterium. You say, well, how is it resistant? It just happens to have a genetic difference than these other ones that makes it less susceptible to being killed by this antibiotic. So you treat with antibiotics, and what happens is it kills all the bacteria, almost all of them, but leaves a few of them alive, and this resistant one, of course, is alive. And so then what happens is the resistant one starts to multiply. And then all of a sudden, all you've got is resistant bacteria in your body. That's the problem. And that's why if you've ever been given a bottle of antibiotics, they will always say, make sure you take the whole thing. And many people will just, oh, I feel better now. Why bother? What happens then, you haven't wiped out all the bacteria, and the more resistant ones are left, and they begin to flourish. So you're trying to use all the you're trying to use all of this, keeping it going, to take the more resistant ones and get them killed as well. All right? Because then they start to propagate, and that's the problem. Just a quick reminder, this organization is run totally by volunteers, but we do have to pay for the production work. If you could help us out by going to give.jesusandscience.org, we'd appreciate it, or you can click in the description box below. If you can't give, we certainly understand, but please just give us a thumbs up and click subscribe. Thank you. And here's the gap. So if you look at the number of antibiotics that have come on the market, new antibiotic classes, were totally new molecule classes. You know, it was really big. They were developing lots of these. Look at this. None. And you say, why are there no new antibiotic classes coming on the market? Well, there's multiple reasons for that. One of them is purely a monetary thing. There's not much money in antibiotics because people sort of want it for free. They don't want to pay for it. You say, well, those mean drug companies. So remember, it takes like $1.5 billion to bring something to market. So how can a company afford that? That's a lot of money. We're not talking million, we're talking billion, $1.5 billion. And so if people are not willing to pay for it, it doesn't justify them going through this. And people say, well, they should just give it away. But the drug company will not exist if it keeps having to make drugs that it won't get money for. That's the problem. All right? And so, and then the other thing is, antibiotics make very little money because after a few years, the bacteria become resistant to it. So you've only been able to use the drug for, say, 5 years. You haven't accrued back all the money you spent bringing this to market through all the testing and everything. And so there's not a monetary incentive to do this because you're fighting these bacteria that keep coming up with new mechanisms to protect themselves. And so what we did is we introduced the molecular nanomachines into this area. And why is this different? It's different because it's not a normal kill. There are ways that people use to kill bacteria, but this one is mechanical. It is actually a mechanical action at the nanometer scale. This is something that is not seen in medicine, in biology. It's not seen in biology. Biology has never seen a drill. Now, if you could fight against a scalpel, you could fight against this, but scalpels, cut stuff right out. So this is now killing with this sort of action and this mechanical action. So what the workers do, and this is actually, this testing has been done by Anna Santos in our laboratory, and she'll test all these different combinations. So DL stands for this is the initials of the postdoc who made the molecule, and this was the molecule number that was assigned to it, meaning the page number within his book that this molecule got assigned. And so you see some of them aren't killing very well, others of them are killing much better. This minimum inhibitory concentration that was needed. And so you go through and you test a lot of these things. And so it turns out then what you do is you test different bacterial strains, E. coli. Many of you have heard of E. coli, for example. And you test them against known antibiotics. So here's novobiacin. Here's novobiacin, and here's how well novobiacin is killing. In other words, you look time and the log. So this is a log scale of how much the cells, these bacterial cells, are dying. The bacterial cell count over the original number. And so you see novobiacin is not working very well. But just the light alone can do something, but it's taking 15 minutes. There are others that are doing this 5 logarithmic reduction in bacterial concentration over a period of 3 minutes. So these are much more active. So we have many of them that are much more active. And again, for E. coli, again, these are much more active. And these are killing both gram-positive and gram-negative bacteria. Gram-negative bacteria have it has a lipid bilayer, and it has a peptides with so it'll have a fat, and then it'll have some sugars hanging off, and then it'll have another lipid bilayer. And so it has these 2 layers. The gram-positive will still have that fat that has the lipid with the sugar moieties on the end, and then the other lipid bilayer. So the gram-negative are actually harder to get through. But again, this is killing both gram-positive and gram-negative. And so we have ones here that are really doing very well in killing these bacteria. It's going to be very hard for a bacterium to find a mechanism to stop this, to evolve a mechanism that can stop this. And that's because this is a mechanical action, something that is mechanically breaking through. It's not a chemical action. Most drugs will chemically do something. And most of them are that are light activated are generating reactive oxygen species, radicals that are ending up killing these. Others do other things where they destroy the membrane layers. This is a mechanical action. And so even the persister cells, there are cells that just are persisting for one reason or another. They are persisting. So sometimes persister cells, they remain longer than other bacteria just because their metabolism is slower. When their metabolism is slower, they're not taking up as much material, so they don't get as much of the drugs into them, and so they just persist longer. These persister cells, it's killing antibiotic-tolerant persister cells. And again, you see the standard treatments. So you're comparing against standard treatments here, and you're seeing much more cell death, much faster, and this is a logarithmic scale in the number of bacteria that are being reduced, the bacterial counts. And then the other thing is that they get these biofilms, these bacterial films that form. And these bacterial films are very hard to get through. So they'll come with these bacterial films here. And what they do here is in these little communities, they're really insidious. They share DNA with each other. And so if you have a resistant cell, it is sharing its DNA with the other cells in these biofilm environments. And that's why in these biofilms, you can start accumulating a lot of bacterial growth. It's first of all hard to get through the biofilm. And second of all, within this biofilm, they're like having a party. And they're just exchanging DNA with each other such that the resistant ones are now becoming they're multiplying in resistance. And again, here's a persister cell. Here's the persister ones. They're slow growing, so they're not taking in as much of the drug, and they end up persisting. And then in these biofilms, it's these persister cells that are sharing their DNA with other cells and making a whole community, in this case, a persister bacteria. And visible light activated and disrupt the biofilm. So what happens is, with this mechanical action, it drills into the biofilms. So you can drill right into the biofilm and start hitting the cells in there. So that's generally very hard to do, to deal with biofilms. But here it drills right into the biofilm. This is going to be a very important advance. One day, it might be that you or one of your loved ones is treated with these molecular nanomachines. Because if we don't find a solution to these, it's just going to be this, and they call it the nightmare scenario, where these bacteria start taking charge. And COVID ended up making it much worse, because what happened is so many people were treated with antibiotics during COVID that more antibiotic-resistant strains came around. So the more people you treat, and that's why physicians are encouraged not to be very loose with giving out giving out antibiotics, because then the strains that are out there are ending up persisting. They want people to see, can you fight this thing off by yourself? Can you fight it off without giving you these drugs, so that we don't get these antibiotics resistance so much? What you can do in a laboratory is you can make antibiotic-resistant bacteria. You intentionally do it. And so you take your parent culture and you grow them, and you see some of them die, but some of them don't die. You just take the ones that don't die well, and you multiply them, so that you get whole populations of resistant bacteria. And so that's what Anna has done. So you make a whole population of resistant bacteria, and you're like, well, that's not good. What if that gets out? Well, you don't let it out. I mean, you got to do these sort of studies, and you're just very careful on how you handle these things. And so you can see that if you look at the typical, like tetracycline was a very popular antibiotic when I was a little kid. But you see that these persister cells, it's not affecting them at all. But in our light-activated nanomachines, and here's polymyxin B. Again, it's not doing very well, but ours are just all baseline. I mean, nothing is getting past these. It's killing them all. When Anna Santos and Brandon go into the lab to work on this stuff, I mean, the bacteria are just shuddering. I mean, they know that their lives are going to be short. And remember, if you have a bacterial population, it doubles every 20 minutes. I mean, they're just growing like crazy. So the population doubles every 20 minutes. So that's why you can feel fine, and an hour later you're like, you've got a fever. I mean, how does that happen? I was feeling fine just a little while ago. Well, because it doubled and doubled and doubled again in that 1 hour. That's what happens with these things. So you can see with the light-activated nanomachines, they're not able to deal. They can't, even though they're trying to build up a resistance, and you genetically keep picking out the ones that have resistance. Again, they can't deal with it. It just kills them. Just kills them. This is why that is so important. Yes?
I have a question. Do you know what's the most common route of transmission for these very resistant bacteria? Like, is it airborne or?
Some of them are airborne. I mean, there's I don't know, how many does Anna test? I mean, probably hundreds of different ones. Some are airborne, so somebody sneezes and you got it. And others go by transfer between bodily contact. There's many in tap water, but unless you have a break in your skin, it's not a problem. And so it's a break in your skin. people that have gone swimming in the ocean where they have an open wound. That's really dangerous because the oceans are full of this stuff. And if you have an open wound, then it becomes really dangerous. So if you have an open wound, you don't want to go swimming in the Gulf of Mexico. Seriously, you don't want to do that. And then that's a real problem.
And I imagine, like, if you were to ingest one of these bacteria, the hydrochloric acid in our stomach doesn't kill them, right?
By ingestion? Yeah, the mechanisms of transfer, well, the acid will kill some of them. But before it even gets there, you're in trouble. You know, your whole esophagus can be lined, and they go in, you know, that way.
Oh.
Yeah. So anyway, so you can see where these things have just drilled in, and they don't just stop drilling once they get through the membrane. They just go in and they just start making jello out of these things. So you can see you can actually visualize the damage. This is on E. coli. Here's 405 nanometer light on E. coli. It doesn't do anything to it. But here, once you have the nanomachine in there, you see these E. coli are just shriveling. And if you look at them up close, they're just pocked with holes all in them. Here's what normal E. coli looks like. And this line is where they're getting ready to divide. So they're just going to divide and keep splitting in half. And these are all, you know, punctured in. So you can actually see a physical damage as well. You can't see the molecules. The molecules are much smaller than this, billions of times smaller than this. But they're leaving holes in there because they've opened up holes in there, and that's what's making these all pocked like this. So these visible light nanomachines, they display broad spectrum antibiotic action. Gram positive, gram negative, they don't care. They disrupt biofilms, they eradicate antibiotic tolerant persister cells, they reduce the MIC of antibiotics with different mechanisms of action. So in other words, what we've done, and this was also some work with work that we did at Texas A&M University, and work that we've done with other groups around, that Anna Santos has done a lot of this work where you can take outdated antibiotics. In other words, antibiotics that don't kill bacteria anymore. And if the bacterium, its mode of action for protecting itself from this antibiotic is to keep that antibiotic from getting through its membrane, what happens is once we add the nanomachines, just a little bit of nanomachines, now those drill holes and the antibiotics go in, and then it ends up killing what would have been a resistant bacterium. So in other words, it potentiates and it synergizes with the outdated antibiotics. Now, if the antibiotic became outdated because it got into the bacterium, but the bacterium right away recognized it and spit it back out, then it can't solve it. It gets in, it gets spit back out. But if it was because it couldn't get through the membrane, boom, it opens up the membrane. So we've got ones that the antibiotics, for example, only work on gram-positive bacteria, but they can't get through the much thicker, more difficult double layer of the gram-negative bacterium. Well, we can take gram-positive action antibiotics, put it with a nanomachine on the gram-negative, open up the gram-negative, the drug gets in, kills. So we can take ones that are exclusively for gram-positive and apply them to gram-negative. And so it gives longer life to even standard antibiotics. And it involves the mechanism of action, the MOA, involves membrane permeabilization followed by leakage of intracellular components and dissipation of membrane potential, resulting in cell death. So what happens is these drill holes, the blebbing occurs, it spews out its guts. All right, and then the next generation is the molecular machines for near-infrared action. So it's a total redesign. What's nice about near-IR light is it has very good penetration through skin. Remember we said you put your hand up to the light, red light gets through. So infrared light gets through. So this was the total redesign. You know, some people say you can even get 10 centimeters of penetration, even 13 centimeters. This is on the very high side. Yes, you'll get some photons coming through 10 centimeters, but at 10 centimeters you've absorbed a lot of photons, and it depends on the type of tissue you're going through. The more water you have, the more near IR light is absorbed. So in tissue that has higher water content, the more that will inhibit the light from going through. It goes through, but just not as much. And so, for example, breast cancers, breast tissue has a lot of water in it, so penetrating through that is harder. But you can certainly get 5 centimeters, which is really quite deep. So the way these work is if you just have a molecule and you excite it, if you heat up the molecule, the bonds start vibrating, but they're vibrating non-concertedly. But there is a way to get whole molecule action, so you can cause the whole molecule to start stretching. So you get all the molecules together start stretching. If you just take a typical molecule and you shine a wavelength of light on it, say 1700 inverse centimeters of infrared light, you'll get the carbonyl, the CO double bond, to start stretching. The other bonds aren't stretching. Remember, all of these are quantized. But if you hit it with there is a wavelength of light that will cause the whole molecule to start stretching if it's a particular type of molecule and if you're using the correct wavelength of light. This phenomenon is called a molecular plasmon, so nanoparticles were known for many years to form plasmons, where you shine light on a say a metal nanoparticle, and the electrons start shifting back and forth to the different sides. So it gets electron rich here and then slides back, electron rich, and you get these plasmons occurring in a nanoparticle. It was just a few years ago that these were hypothesized to happen with molecules too. And that effect was first hypothesized by a professor here at Rice in the physics department, and he was the first to hypothesize that a molecule could undergo a plasmon. And so what would happen is that he said that if you had certain types of molecular designs, they could shift the electron density back and forth. You may remember when we talked about carbon nanotubes, we talked about the radial breathing mode, that the whole nanotube would just start breathing, expanding like this when you hit it with a certain wavelength of light. That is a plasmonic mode, but that's in a nanometer-sized structure, not just a tiny little molecule. So you see this stretching effect in both dimensions. That's what we're going to capitalize on. So there are these type of dyes called cyanine dyes. And these cyanine dyes, they have a plus charge on one side and a neutral side on the other. And if you just take these two electrons, you push them, these shift, all the way to this becomes neutral, this becomes positive charge. So cyanine dyes have this ability to move charge back and forth by resonance formation. When you shine light on these will become a molecular plasma, and they'll start to stretch like this and like this. And it turns out my student recognized where the plasmonic mode would be hit, and it's the vibronic mode. And this is where the vibrational mode of the molecule couples with the electronic mode. You get electronic excitations, you get vibronic excitations. When the two of them couple, that's called a vibronic mode. This was known in nanoparticles. People had taken molecules and they excited them with STM tips to do this. He was the first to recognize that this little hump here on the higher energy side, the lower wavelength, higher energy side of the lambda max. So here's the lambda max, but right here. And then he used a light that gives this red, so that it's exciting right on that mode. So he found a light with the correct wavelength to excite right on that mode. And that was Cicerone Ayala Orozco who found that. The idea is that these molecules will sit in a lipid bilayer. You shine near IR light on them and they just start jackhammering, just start stretching. And that just breaks up the membrane. And you're going to see here, if this movie runs, I want you to watch right here. You see, there's molecules. The molecules have some inherent fluorescence. They're fluorescent, so that's very convenient. You don't have to put a fluorescent dye attached to them. So, and they're stuck, a lot of them are stuck in this lipid bilayer, right? These are the lipid bilayers of the cells. So just watch right here. And so the DAPI dye is getting into the cell and turning it blue. And you see the lipid bilayer is gone. So this particular dye, DAPI, will turn blue once it gets into the cell. So it's gotten in, and even the whole membrane you could see is just gone. It's just gone. This is like so exciting. He came up with a totally new molecular structure that does this, and it's near IR active. You say, well, why don't you just shine near IR light on the pharyngomotors? Because it doesn't excite them. It doesn't excite them. We had to go to a totally different molecular design, and this was come up with by the student on the project, the postdoc. And so you can see here's this mode that he's going to excite right here. And he looked at the different molecular orbitals and how those would map out and what was going to stretch here. And so a lot of this, one of my former postdocs was a professor at A&M. He did the theory on this. Again, it was Cicerone that did the main part of this. And then Professor Myers and Rangel are at MD Anderson. So we're working in their lab along with their team on this. And so you can see the effects of this. And then in other papers, we were doing other theory that was done by Professor Kolomeski here at Rice and his students. And so you can compare. Remember we said this spins at 3 million rotations per second? These oscillate at 10 to the 13 oscillations per second. So it's 10 to the 7th. So that's 10 million times faster than the pharyngomotors. 10 million times faster. So this is pharyngomotor is spinning at 10 to the 6th rotation. This is oscillating at 10 to the 13 oscillations per second. This is so fast. It is so fast. It's like sonicating the outer membrane. But this is just lightning fast. These start oscillating. And this is going to be a tremendous effect. This effect of these molecules acting like this is going to translate not just for killing cancer, which you're going to see, but for killing superbacteria. It's going to translate for other things that you want to do in a cell. You associate these molecules with, say, DNA and get it vibrate, boom, it'll break it open. We're associating these molecules and we're causing it to activate calcium release. Maybe we'll look at that next time, maybe not. I haven't chosen that yet. But and also stimulate muscle movement through this oscillation of molecules. So it's 7 orders of magnitude faster than pharyngomotors. So you can see what happens, that if you just take the size 7.5 amine and you put it with the cells, nothing happens. If you take DMSO with cells, nothing happens. DMSO and light, nothing happens. You take the size 7.5 amine, one of these molecular jackhammers, and light, boom, the cells are just gone. Just gone. Here's cell viability. It's just gone. I mean, it just kills them all. Just kill them all. Just wipe them out. And this is melanoma cells, skin cancer. And so some people may say, well, this is just photothermal therapy. Yes?
I don't understand how we're not killing normal cells too.
Oh, you're killing normal cells. You're killing normal cells, but you inject this into the tumor. And if there's normal cells, it's collateral damage, which occurs in battle, you know? And but the same thing happens when you excise melanoma. I'm sure you've never had it done. I've never had it done, but particularly people as they get older and people with lighter skin have a lot more problem with it, and especially men who are bald have a lot of problems on their head. And so what happens is you go to the dermatologist and they cut it out. And they go much deeper than it needs to be. So they're taking all sorts of good tissue with it, just to make sure that none reaches down and hits the lymph system, which we've looked at before. So yeah, it's hitting good stuff. But every cancer drug you take is still hitting good. That's why people get really sick. If it could just go. Now, the way you target these is you put a peptide on them to go to a particular cancer cell type. So this has a double mechanism of safety. You can target these to a particular cancer type, and you only shine light at that point. So all the stuff that's in cells, it's like nothing's happening. Only what gets the light shown on it. So it's it's doubly safe. It's much safer than you're just taking a drug and it's killing good cells, killing bad cells. And that's why cancer patients get so sick, because all their good cells are getting killed too. And you just hope that you kill more cancer than the good cells. But so this is killing good cells as well. So what he did is he cooled this thing down, and he showed that it was not photothermal. It was not because the sample is heating up. You could do the same thing in ice water. It's going to do the same thing. So it's not from a cooling effect. And it's not photodynamic therapy. It is not the ejection of free radicals that causes this, because you can put free radical inhibitors in here and it'll stop that. It won't stop that from happening. It'll still kill the DAPI positive cells. And this means the cells that have taken up the DAPI, the cells that have died now. So it's just like an inverted plot. But you put different inhibitors in here, and still it doesn't stop these things. It just keeps going. And then what he showed here, this is really interesting. So you take a lipid bilayer and you associate it with these little molecular jackhammers. Now you shine a light. What do these jackhammers do? They throw out the lipids. They start shaking and the lipids just fall out. And when they fall out, it causes a contraction now of the lipid bilayer, because it's thrown out all these lipids. And then you do it again and it'll cause contraction to, at some point, it can't get any smaller, the thing just explodes. And so you can see here, here's the Cy5 amine. So here's the bright field, here's the fluorescence field. So this is a liposome. This is a lipid bilayer, just like just the lipid bilayer part, not really a cell, just lipids forming this bilayer in a sphere. You know, we're looking at it just as a ring, but remember, this is a sphere. And now what happens is when you shine light with those molecules, look what happens. It gets smaller, smaller, boom, it's gone. Just by throwing out, it keeps throwing out lipids till you get this small, and after that you hit the critical size, it can no longer exist, and it just breaks up. And so here's the treatment of mice, and so you can see that it's killing these tumors. Here's the tumor growth after 25 days. It's like 2,300 cubic millimeters, so a big tumor on the flank of that mouse. And then you just gave 4 treatments, and the tumor growth is much smaller. So this was on this B16F10 melanoma. Here's this A375 tumors. And so again, it has just eradicated the tumors. So here's tumor growth unchecked. Here's the tumor growth. It's just eradicated it. And so what happens is, this is a survival plot. You want to see how many of these animals survive this. And typically, all the animals will die from these tumor cells on them. All the animals die. But here, 50% of the animals live. And we track them out 7 months. Nobody tracks them out 7 months. A mouse lives 2 years, full lifetime. So to track 7 months is a long time in a mouse, and they're still not dying. It eradicated the cancer, so that even in the title of the paper, the editors allowed us to use the term eradicated cancer cells, which is a very strong term. But half of the mice, and this was not even an optimized dose. This was just the first run through that we did this. So it's a very powerful technique. And so we can study this, we can study different jackhammer types and how effective they are. This is going to be a weak jackhammer at a 7.0 versus a 7.5. The 7.5 has this extra ring up here. See, this doesn't have that extra ring. And you can tell, you can track this, and you can see that it's going to have a much larger effect. And what he did is he figured out a way to determine how effective this stretching mode is. And so he plotted this out. It's just an amazing, amazing study that he's done. And then we made a lot of molecules. We made a lot more than this now, but we've made a lot of different molecules, and this is where the synthetic chemists come in. So they're just in the lab making molecule after molecule. They don't touch a cell. But then there's other people that work with the cells. And then we have students that work with the animals, go up and work with the animals. But you just make lots of different combinations of these, and you see what kinds of groups really cause this to work better, and then you see the good ones and you build around those groups. And now we're using a whole machine learning model, taking all the hundreds of compounds we've made and do machine learning and say, okay, what are the salient features here? What are the features we want for this activity, for cancer, for bacteria? And you start doing this, and you start mapping this out and have AI do that for you. So here's the structure-activity relationship. So you see, here are the ones that are just killing the cancers very well. Here are other ones that are not working as well. But here, these are working really well. So from this structure is up here down to this structure is the optimal structure. And you can see that you just go through these plots and you just test the efficacy of this. He came up with a way to just measure this using the UV spectrum, and you can predict what he called the plasmicity, experimental plasmicity index, the EPI, to detect this. And just wiping out cell type after cell type. So, what's the take-home message? Well, when you go in for treatment of cancer, there's many different ways that you can be treated. You have surgery, where they remove it. You have radiation. What radiation is doing is it's generating radicals in there. Very reactive species, a 7-electron species, which is plucking off things and killing it. So you can have chemotherapy, where you have taking chemicals. You can have immunotherapy, where they try to activate your immune system to go after the cells. You can have genetic therapy, where they'll genetically modify. That's a newer thing, to try to genetically modify the host so that it can battle this cancer. And this adds now a new modality, a mechanical effect at the nanometer scale. It's molecular machines, it's nanoscalpel-like. And then you can couple this with immunotherapy, because you can cause the cells to spit out their guts. Remember, the proteins are around, and then your immune system can act on that and then go find every cell that has that protein. So that's the end of nanomedicine. Are there any questions? Okay, we'll do something else next time. Thank you. If you're enjoying this series, give us a thumbs up and click the subscribe button, and that way you'll hear when we're coming out with new videos. There are no salaried employees in this organization. All the accounting, all the legal work, that's all done by friends of mine. The only thing that I have to pay for is the production work, and if you could help us out with that, I'd appreciate it. There's a link below where you can just click on that and help us in several different ways. Thank you.